Observe this structure. This continuous, undulating surface is a biological cell membrane, keeping the organized interior of a cell isolated from the extracellular environment. For decades, biologists viewed this as a static wall. That changed in 1972, when Singer and Nicholson proposed a fluid mosaic model. They described the membrane as a two-dimensional liquid, a fluid environment where embedded proteins diffuse laterally across the surface. The physical constraints of this system are tight. This selectively permeable barrier is approximately 5 nanometers thick. Across that microscopic span, the membrane acts as a gate for nutrients, while simultaneously serving as a signaling hub, using receptors to detect and relay external data. It handles the physical mechanics of cell division, or cytokinesis, and displays surface molecules that allow neighboring cells to recognize one another. This creates a unique physical challenge. A chaotic, 5-nanometer liquid film must maintain the structural stability required to keep a biological cell alive. This stability emerges from the membrane's role as a tuned thermodynamic machine, where molecular geometry dictates physical performance. The membrane lipid is an amphipathic molecule with a polar head and nonpolar tails. Placed in water, the tail forces surrounding water into rigid cages, dropping entropy. But as lipids aggregate, tails sequester together, releasing water and causing entropy to spike. This spontaneous, entropy-driven process is the hydrophobic effect. This means bilayer assembly is driven by the maximization of water entropy, rather than attractive forces between the lipids themselves. Even when millions of lipids align into a flat, double-layered sheet to hide their tails, the system remains physically stressed. The exposed edges of the flat bilayer still touch water, incurring an energetic penalty. To eliminate this energetic penalty, the flat sheet spontaneously curves and closes in on itself, forming a completely sealed compartment. The fundamental architecture of cellular life follows the physical requirement to avoid these thermodynamic penalties. While thermodynamics dictates the basic bilayer shape, the actual chemical makeup of these boundaries is variable. Ratios of lipids, proteins, and carbohydrates are tuned to support specific survival needs. Using the red blood cell as a baseline shows a membrane split of 49% protein to 43% lipid. Contrast that with a mitochondrion, requiring 80% protein for ATP synthesis. Conversely, the myelin sheath uses 80% lipid to provide electrical insulation for neurons. This infographic details the scaffolding itself. The lipid unit is classified into 3 primary categories: phospholipids, glycolipids, and sterols. Glycolipids, which include gangliosides, are crucial for cell recognition and are localized strictly to the exoplasmic leaflet of the bilayer. Researchers confirm this asymmetry by applying large lectin molecules, which bind to the exterior sugars, but are physically incapable of crossing the internal boundary. Chemical ratios and localized structural molecules directly hardwire the cell's specialized capabilities. To function, these biological structures must maintain a thermal equilibrium, balancing between a solid, crystalline gel phase and a fluid sol phase. The boundary of this equilibrium is the transition temperature, or Tm. It marks the thermal point where 50% of the membrane lipids are in the gel state and 50% are fluid. This phase transition is mapped using differential scanning calorimetry. This animated graph shows a standard DSC thermogram, reaching its peak at the Tm point. That peak represents the heat energy required to overcome van der Waals interactions locking the tails. Looking at the bottom left schematic, the first mathematical rule of transition temperature is shown. Tm is directly proportional to fatty acid chain length. Longer chains require more heat to break apart. The second rule shows Tm is inversely proportional to unsaturation. This table of phosphatidylcholine variance tracks how unsaturation manipulates melting points. DSPC is fully saturated. Its straight chains pack tightly, resulting in the transition temperature of 58 degrees Celsius. DOPC is polyunsaturated, dropping the melting point to -22 degrees. This temperature drop is caused by the kink factor. A carbon-carbon cis double bond forces a rigid angled bend, preventing tight packing and increasing membrane fluidity. This applies only to the cis configuration. Trans-unsaturation produces a straight chain that mimics saturated fats and fails to enhance fluidity. Organisms leverage this math to survive. A wheat plant exposed to cold temperatures actively incorporates unsaturated fatty acids into its cell boundaries. By engineering molecular kinks into its architecture, the plant lowers its Tm, ensuring its cells do not freeze into a non-functional gel state. The fluid mosaic is dynamic, but it is not a random mixture of floating parts. Populating the barrier requires metabolic coordination across multiple compartments, utilizing the endoplasm to construct specialized microdomains known as lipid rafts. These are localized, organized islands drifting within the broader fluid environment. To maintain structure against surrounding fluidity, these rafts are enriched with cholesterol and sphingolipids, stabilizing the domain. By clustering specific molecules together, lipid rafts serve as platforms for complex signal transduction and as focal points for pathogen entry. Cells engineer physical deviations called caveoli. These cup-like membrane invaginations are mechanically maintained by the interaction between integral caveolin proteins and cytoplasmic cavin proteins. The asymmetrical distribution of lipids creates a heterogeneous landscape, providing the physical hardware required for complex eukaryotic life. This diagram aggregates the total molecular architecture of plasma membranes. Maximizing water entropy acts as the thermodynamic engine, forcing lipids into a sealed liquid. Intercalated proteins perform metabolic lifting, while within the core, molecular kinks and sterols tune the phase state against environmental shifts. The membrane maintains its barrier function because of its dynamic fluidity, adapting to thermal shocks and mechanical stress without fracturing. The fluid mosaic is a highly active, thermodynamically calibrated machine, where every carbon bond ensures the continuation of cellular life.